Welcome back to Molecules Matter with Dr. Dan, the podcast where we take one molecule found in the foods you eat and trace it all the way back from its origin in nature to its effect inside your cells. Because here's a core idea that runs through every episode. New molecules create new signals, and new signals create new cellular outcomes, and that leads to a new you. Today we're doing something a little different. We're going to look at one specific molecule, chrysin, and then zoom out to the whole food it lives in, honey. Because honey isn't just sugar in a jar, it's a genuinely complex biological product made by an insect out of plant material, and it carries dozens of bioactive compounds along with it. Chrysin is one of the most interesting of those compounds, and understanding it is a great way to understand honey as a whole. So, let's get into it. Before we get to the molecule, it's worth remembering what honey actually is, biologically speaking. Bees collect nectar from flowers, which is mostly water and sucrose. Back at the hive, worker bees pass that nectar mouth to mouth, adding enzymes, most importantly invertase, which breaks sucrose down into glucose and fructose, and glucose oxidase, which we'll come back to later. The bees then fan the nectar with their wings to evaporate most of the water, concentrating it into the thick, shelf-stable substance we call honey. Along the way, honey picks up trace amounts of pollen, wax, and, depending on the region and the trees nearby, resin compounds like chrysin. That's the whole reason honey isn't just bee sugar. It's a processed, enzyme-modified, chemically active substance, and the specific plants and trees in a bee's foraging range determine a lot of its final chemical makeup. That's why honey from a region full of poplar trees can carry meaningfully more chrysin than honey from a region without them. Chrysin is a naturally occurring flavone, a subclass of flavonoids, which are the pigment and defense molecules that plants produce. Structurally, it's one of the simpler flavones, but simple doesn't mean weak. Chrysin shows up in a surprising number of places. In honey, in small amounts, in propolis, which is the resinous bee glue that bees use to seal and sterilize the hive, in passion flower of the Passiflora species, and in certain mushrooms. Here's the part that makes honey so interesting. Bees don't make chrysin themselves. They collect it. Bees gather resin from tree buds, especially poplar and birch trees. They mix it with wax and their own enzymes and turn it into propolis, which they use to disinfect the hive. Some of that resin and propolis residue ends up mixed into the honey itself, especially in honeys made near poplar-heavy regions. So, when you eat honey that contains chrysin, you're essentially eating a molecule that started as a tree's chemical defense system, got harvested by a bee, processed inside the hive, and ended up in your jar. That's a pretty remarkable supply chain for one flavone. Chrysin has been studied for a wide range of biological activities. The major categories researchers keep coming back to are number 1, antioxidant activity, number 2, anti-inflammatory activity, number 3, antibacterial and anti-allergic activity, number 4, anti-tumor activity, number 5, effects on hormone metabolism, and number 6, neurological and antioxidant effects. Let's go through the ones with the most interesting mechanisms. Chrysin and inflammation. One of the clearer mechanisms for chrysin involves the NF-kappa B pathway, the same master inflammatory switch we talk about often on this show. Research on skin inflammation has shown that chrysin can bind to NF-kappa B kinase, which prevents the breakdown of NF-kappa B. When NF-kappa B stays intact, it holds NF-kappa B in check, which in turn reduces the production of inflammatory signaling molecules like CCL5, a chemokine involved in conditions like atopic dermatitis. In plain terms, chrysin appears to jam one of the body's key turn-on inflammation switches before it fires. This is consistent with chrysin's broader reputation as an anti-inflammatory flavonoid, and it's one of the more mechanistically well-supported effects in the literature. Let's slow down on the antioxidant piece for a second, because antioxidant gets thrown around loosely. Chrysin's structure includes hydroxyl groups that can directly donate electrons to unstable reactive oxygen species, neutralizing them before they damage lipids, proteins, or DNA. That's the direct chemical action. But chrysin also appears to work indirectly by upregulating the body's own antioxidant enzyme systems, things like superoxide dismutase, catalase, and glutathione peroxidase. In other words, it doesn't just mop up damage itself. In some models, it appears to boost your own internal cleanup crew. This dual action, direct radical scavenging plus support for your endogenous antioxidant enzymes, is a pattern we see across a lot of plant flavonoids, and it's part of why they show up so often in inflammation and disease research. Chrysin has been studied extensively in cell and animal models across several cancer types, including breast, prostate, lung, liver, stomach, and melanoma. The mechanisms researchers have identified include inducing apoptosis or programmed cell death in abnormal cells. Number 2, altering the cell cycle, slowing abnormal cell division. Number 3, inhibiting angiogenesis, which is the formation of new blood vessels that tumors rely on to grow. And number 4, reducing invasion and metastasis in lab models. In melanoma cell studies, chrysin has been shown to increase apoptosis-related proteins while also triggering autophagy, essentially activating 2 separate cellular cleanup mechanisms at once. In lung cancer cell models, it appears to work partly by inhibiting a signaling protein called AKT, which many cancers rely on to survive. Now, I want to be very clear about something here, because this is where headlines tend to run ahead of the science. This is overwhelmingly cell culture and animal data, not human clinical trial data. That doesn't make it meaningless. This is exactly the kind of early-stage research that identifies promising compounds worth studying further. But it does mean we should describe chrysin as a molecule with anti-cancer research potential, not as a treatment at this point. If you spend any time in the supplement world, you've probably heard chrysin mentioned in the context of boosting testosterone. Here's where that claim comes from. Chrysin has been shown in laboratory studies to inhibit an enzyme called aromatase, the enzyme responsible for converting testosterone into estrogen. In vitro, chrysin is actually one of the more potent aromatase inhibitors, flavones, that have been tested. That sounds like a direct path to less testosterone converted to estrogen equals more testosterone available. And that's exactly the logic that made chrysin popular as a bodybuilding supplement in the early 2000s. But here's what actually happens when researchers tested it in humans. A human study gave men in their mid-20s honey and propolis tablets containing chrysin daily for 21 days and measured urinary testosterone levels before and after. Urinary testosterone levels did not significantly change. Well, why the disconnect between the lab data and the human data? The leading explanation is bioavailability. Chrysin has very poor water solubility and gets metabolized and eliminated quickly once it's absorbed, so very little of it actually reaches circulation in an active form when taken orally at normal doses. The aromatase inhibiting effects that looks impressive in a test tube largely doesn't translate into the bloodstream at doses achievable through food or standard supplements. This is a really important lesson that applies far beyond chrysin. A mechanism that works in a petri dish only matters if the compound can actually get into your body in a meaningful amount. There's a deeper reason for this too. Once chrysin is absorbed in the gut, it gets rapidly tagged by liver enzymes through a process called glucuronidation. Essentially, your body attaches a sugar acid molecule to it, marking it for elimination through urine. This happens so efficiently that only a small fraction of ingested chrysin ever circulates in its active, unmodified form. It's one of the most well-documented bioavailability problems in flavonoid research, and chrysin is often used as the textbook example of it. There's also a body of research on chrysin's effects on mood and anxiety. Chrysin has structural similarities to compounds that interact with GABA-A receptors, which is the same receptor system targeted by anti-anxiety medications. Animal studies have explored chrysin as a natural anxiety-reducing compound because of this pathway. This research is still preliminary and mostly limited to animal models, but it adds another dimension to chrysin's profile. It's not just an antioxidant and anti-inflammatory molecule. It may also have direct signaling effects on the nervous system. Like most flavonoids, chrysin's hydroxyl groups allow it to neutralize free radicals directly, and studies have also shown antibacterial and anti-allergic activity, like contributing to some of honey and propolis' traditional use for minor infections and skin irritation. Chrysin is a genuinely interesting molecule with a wide range of laboratory-demonstrated activities: antioxidant, anti-inflammatory, antibacterial, anti-allergic, and potentially anti-cancer and anti-anxiety effects. But the amount of chrysin naturally present in a spoonful of honey is small, and its bioavailability is limited. So, while chrysin is a great window into how plant and hive chemistry can influence human biology, don't expect a jar of honey to meaningfully shift your hormones or treat disease on the strength of its chrysin content alone. Which brings us to the bigger picture, because chrysin is only one piece of what makes honey worth talking about. Honey contains hundreds of compounds beyond chrysin: other flavonoids, phenolic acids, enzymes, organic acids, and unique sugars. A lot of honey's benefit comes from this entire matrix working together, not any single molecule. Honey's antibacterial activity comes from several overlapping mechanisms. Number one, hydrogen peroxide production, generated when an enzyme B's add to the honey, which is called glucose oxidase, when it reacts with glucose and moisture. Number two, low water activity and high sugar concentration, which draws water out of bacterial cells through osmosis. Number three, acidity. Since honey typically has a pH between 3.2 and 4.5, this pH is hostile to many pathogens. In Manuka honey specifically, an additional compound called methylglyoxal, which provides antibacterial activity independent of hydrogen peroxide. Research on medical-grade honey has found it to be effective against a wide range of bacteria, including several multi-drug-resistant strains, which is part of why honey-based dressings are used clinically for certain wound care applications. This is one of the best supported uses of honey outside of the kitchen: wound healing. Clinical evidence shows that honey dressings can heal second-degree burns several days faster than some conventional dressings. And there's moderate quality evidence that surgical wounds that become infected can heal faster with honey-based treatment. And there's an important caveat here, though. This applies specifically to sterilized medical-grade honey used under medical guidance, not honey straight from your pantry applied to an open wound. Honey has real evidence behind it as a cough remedy, particularly in children. Multiple studies have found honey performs comparably to or better than some over-the-counter cough suppressants for reducing cough frequency and severity, likely through a combination of its soothing, coating texture, and mild antimicrobial action on the throat. Beyond chrysin, honey contains other flavonoids and phenolic compounds. The darker the honey, generally, the higher its total antioxidant content. Darker honeys, like buckwheat and manuka, tend to outperform lighter honeys, like clover, on this measure. Honey contains oligosaccharides, which are complex sugar chains that aren't fully digested in the small intestine and can serve as fuel for beneficial bacteria in the gut. This is an area of ongoing research, but it suggests honey may have a small supportive role in gut microbiome health beyond its simple sugar content. If you've shopped for Manuka honey, you've probably seen labels like UMF or MGO with a number attached. These ratings measure the concentration of methylglyoxal, which is the compound responsible for Manuka's extra antibacterial punch beyond what regular honey provides. Higher numbers mean higher methylglyoxal content and, generally, a higher price tag. For everyday use, this level of potency usually isn't necessary. It matters more for the medical-grade wound applications we just discussed. So what about the local honey cures allergies claim? This is one of the most common things people tell me, so let's address it directly. The theory is that eating local honey exposes you to local pollen in small amounts, building tolerance the way allergy shots do. It's a reasonable sounding idea, but it hasn't held up well in controlled studies. The pollen that actually causes seasonal allergies is wind-pollinated grasses, ragweeds, trees, and bees rarely collect it, since bees are after nectar from insect-pollinated flowers. So the pollen load in most honey doesn't overlap much with what's triggering your allergies in the first place. That doesn't mean honey has zero relevance to allergies, but the local honey builds tolerance mechanism specifically doesn't have strong evidence behind it. Let's talk about what honey is not. I'd be doing you a disservice if I didn't balance this out. Honey is still, nutritionally, mostly sugar, primarily fructose and glucose. It raises blood glucose, and people managing diabetes or insulin resistance need to treat it the same way they treat any other concentrated sugar source, not as a health food loophole. There are two real safety notes here. Number one, never give honey to infants under 12 months. Honey can contain Clostridium botulinum spores, which an infant's immature gut can't yet defend against, risking infant botulism. This isn't a risk for older children or adults with a mature gut microbiome. And the number two safety note, if you have pollen allergies, raw honey occasionally causes reactions since it can contain trace pollen and propolis residue. So let's talk about how to actually use this information. Practically speaking, if you want the broadest antioxidant and phenolic content, choose raw, darker honeys like buckwheat, manuka, or wildflower honey over heavily processed light honey. Use honey as a sugar swap in moderation, not as an added supplement on top of your existing sugar intake. For a sore throat or occasional cough, a spoonful of raw honey or honey stirred into warm tea has genuine evidence behind it. Don't rely on honey or chrysin supplements for hormone optimization. If you're interested in that goal, that's a conversation for a doctor, not a spoonful of honey. Keep it away from children under 1 year old. Full stop. Now, my closing thoughts. What I love about this episode is that it shows both sides of how we should think about molecules in food. Chrysin is a legitimately fascinating compound. It's a tree's defense molecule harvested by bees with real laboratory demonstrated effects on inflammation, cancer cell behavior, and possibly the brain. But its low bioavailability means the honey on your counter isn't delivering a therapeutic dose of it. Meanwhile, honey is a whole food. Sugars, enzymes, other phenolics and all has genuinely useful, well-documented properties for wound care, cough relief, and antimicrobial action that don't depend on chrysin at all. That's the nuance that matters. Not every benefit of a food traces back to its most famous molecule, and not every famous molecule delivers on its lab-based promise once it's inside a real human body. Your body doesn't care about marketing claims. It responds to what actually reaches your cells, in what amount, and how often. That's the power of paying attention to the molecules, not just the labels. And that's why molecules matter. If you enjoyed this episode, make sure to, number 1, follow the podcast. Number 2, share it with someone who reaches for honey when they're sick. And number 3, leave a review. It helps more people discover this content. You could also find more episodes and resources at www.drdangoobler.com. Thanks for listening, and I'll see you next time.